morning one and all. I am Dr. S. Marina Prem Kumari, working as Professor Biotechnology at Agriculture College and Research Institute, Madurai. And today I am going to take on the topic concepts, history and scope of plant tissue culture. And first of all, we will see some of the concepts of plant tissue culture. And in this one, first we will see about what is plant tissue culture. So plant tissue culture means it is growing of plant cells, tissues or organs in an artificial or synthetic medium under controlled condition is called plant tissue culture. So next we will see about the plasticity. So plasticity means plants generally they have the nature of having a long life cycle and they lack the mobility. So due to lack of this mobility, they have to survive to the extreme environmental conditions. And this capacity to survive the extreme environmental conditions is called as the plasticity. And next we will see about the totipotency. So totipotency is the potential of the plant cells or tissues to develop into a whole plant. So in the presence of correct physical as well as the chemical stimulus, that is called as the totipotency. Next is the recalcitrant. An explant is said to be recalcitrant if it is difficult to give rise to an organism or whole plant. So explant means it is a plant organ or piece of tissue used to initiate the culture. So that is called as the explant. Node. So it is the region on the stem from which a leaf bearing or an axillary bud arises. So morphogenesis means it is the anatomical or physiological events involved in the regeneration process, and in this regeneration process it undergoes dedifferentiation and redifferentiation processes. Meristem, it is the localized group of actively dividing cells in any explant. Meristemoid, it is the localized group of meristematic cells that arise from the callus, and it may give rise to the organs like roots or shoots or both the root as well as the shoot system. Next it is the differentiation. This occurs normally in the plants. The meristematic tissue is differentiated into simple or complex tissues, that is called as the differentiation. And these are the important concepts in the case of plant tissue culture, that is regeneration. So it is the morphogenetic response that results in the formation of new organs like a shoot or root, and also embryos or whole plants from the cultured explants. So dedifferentiation, it is the phenomenon of mature cells reverting to meristematic state to produce the callus, and this is called as the dedifferentiation. Redifferentiation means the ability of the callus to develop into a plant organ or a whole plant is regarded as dedifferentiation. So this is the diagrammatic representation of the process of regeneration. So explant we have to inoculate into the artificial synthetic medium, and this undergoes dedifferentiation to form the callus, which is nothing but the undifferentiated mass of cells, and these callus they redifferentiate to form the plants. So these plants they have the differentiated cells either through organogenesis or embryogenesis. So below is the example of a leaf explant which is inoculated and that produces the callus, and from callus there is the plant regeneration through the process of dedifferentiation and redifferentiation. So adventitious, it's an important term which is used in plant tissue culture, that shoots or roots that arise from the leaf or stem tissues apart from the axils or the apex, and also the embryos that form apart from the zygote. So these are called as the adventitious buds. Next is the determined cells. So these cells are committed to a particular pathway of development to form any differentiated organs. So these are called as the determined cells. Heterotrophic means it is if the explant is dependent on external energy source and also the external cultural conditions, then it is called as the heterotrophic. It is just the opposite of autotrophic nature. Next is the polarity. So the entire plant is bipolar in nature, that is it has got two ends. One is the plumular end where the shoots develop, another one is the radicular end where the roots develop, and apart from that there is two more regions that is called as the distal as well as the proximal end. The distal end is the region which is apart from the point of attachment, and in the case of stem, and in the case of leaf, or in the case of leaf, stem as well as root also, it is the distal portion that is called distant portion, that is called as the distal end. Another one is the proximal end which is nearest to the point of attachment. So these are also the important terms in the case of tissue culture. Now we will come into the important discoveries in the case of plant tissue culture. First of all, in 1756, that is Duhamel du Monceau, during his studies on the wound healing in the plants, he observed the callus formation. So this was the first time this callus was observed. In 1838, totipotency theory was formulated by Schwann and Schleiden. So this is the principle where the plants or the explants any part of the plant is able to develop into a complete plant, that is called as the totipotency. In 1892, plants synthesize the organ forming substances which are polarly distributed. So organ forming substances means it is the auxins as well as the cytokinins which are distributed in the polar region. So that was discovered by Sachs. In 1902, first attempt at plant tissue culture was done by Gottlieb Heberland. He is the father of plant tissue culture. In 1904, first attempt at embryo culture of selected crucifers was done by Hanning. In 1909, fusion of plant protoplast, although it failed to produce survive, it was done by Kuster. In 1922, in vitro culture of root tips was done by Robbins. In 1925, embryo culture was applied in the interspecific crosses of linum by Leibeck. In 1934, successful culture of tomato roots was done by White. In 1939, there was the successful culture of the callus culture. So that was done by Goddard, Nobicott and White. In 1940, in vitro culture of cambial tissues of Ulmus tree, that is cedar elm tree, to study the adventitious shoot formation was done by Goddard. In 1941, this coconut milk, that is which contains the cell division factor, mainly it is the cytokinin, was the first time used for the culture of datura cells by Van Overbeek. In 1941, in vitro culture of crown gall tissues was done by Browner. And in 1944, first in vitro culture of tobacco explants was done to study the adventitious shoot formation, that was done by Skoog. In 1948, formation of adventitious shoots and roots was observed in tobacco, and it was determined by altering the ratio of auxin and adenine. That was done by Skoog and Sui. In 1950, organs regenerated from the callus tissues of redwood tree, that was done by Ball. And 1952, virus-free dahlia plants was developed by Meristem culture by Morrell and Martin. And then 1952, first application of micrografting was done by Morrell and Martin. Micrografting means that is the union of rootstock as well as scion inside the tissue culture tubes in the artificial medium, that is called as the micrografting. In 1953, haploid callus was observed from the maiden hair tree, which is produced from the pollen grains. That was done by Tulecki. And 1957, discovery of regulation of organ formation roots and shoots by altering the ratio of cytokinin and auxin was done by Skoog and Miller. In 1958, regeneration of pro-embryos from callus clumps and cell suspensions of Daucus carota was done by Reinert and Stewart. So this is an important discovery in the case of tissue culture. So regeneration of pro-embryos from the callus, that is somatic embryogenesis. In 1959, publication of the first extensive handbook on plant tissue culture was done by Goddard. And in 1960, enzymatic degradation of the cell wall to obtain the large number of protoplasts was done by Cocchi. And 1960, vegetative propagation of orchids by Meristem culture was done by Morrell. And 1960, filtration of cell suspensions and isolation of single cells by plating was done by Bergman. That is called as the Bergman plating method. And 1962, the development of the famous MS medium, that is Murashki and Skoog medium, was done by Murashki and Skoog. And 1964, first haploid datura plants was produced from pollen grains by Guha and Maheshwari. These two persons were the Indian scientists who had discovered the haploid plants from the pollen grains. In 1964, regeneration of shoots and root on callus tissue of populus, that is white poplar tree, was done by Mathis. In 1965, differentiation of tobacco plants from single isolated cells in microculture. So microculture means that is it may be a single cells, 2 cells or 3 to 4 cells, that's all. So very limited number of cells are cultured in Petri plates, that is called as the microculture. that was done by Vasil and Hildebrandt. In 1970, selection of biological mutants in vitro was done by Coulson. In 1970, again first achievement of protoplast fusion was done by Power et al. Protoplast fusion is mainly for developing the somatic hybrids. In 1972, interspecific hybridization through protoplast fusion was done in 2 Nicotiana species, that is by Coulson et al. In 1972, induction of axillary branching by cytokinin in the excised Geribera shoots was done by Murashki et al. In 1974, regeneration of haploid Petunia hybrida plants from protoplast was done by binding. 1974, fusion of haploid protoplast found possible, which gave rise to hybrids, and this was done by Melchers and Labib. 1974, biotransformation in plant tissue cultures was done by Reinert. So, biotransformation means it is transforming the secondary metabolites. Secondary metabolites are produced by cell suspension culture, and these secondary metabolites can be transformed into novel compounds by altering the chemical nature, by adding some groups into it. So, that is called as the biotransformation. In 1974, discovery of this TI plasmid, tumor-inducing plasmid, was done in the by Agrobacterium tumefaciens. That was done by Zenon and Lerbecki. In 1976, interspecific plant hybridization by protoplast fusion of Petunia hybrida and Petunia paradoi was done by Power et al. In 1976, successful integration of TI plasmid DNA from Agrobacterium tumefaciens into the plants was done by Clinton et al. In 1978, somatic hybridization of tomato and potato was done by Melchers et al. And then 1979, co-cultivation procedure for transformation of plant protoplasts along using agrobacterium was done by Martin et al. In 1981, introduction of the term somaclonal variation by Larkin and Scowcroft. Somaclonal variation means when culturing the plant tissues in an artificial synthetic medium or tissue culture medium, so due to the repeated subculture, they are able to produce the variants. So, these are called as the somaclonal variants. In 1982, fusion of protoplasts by electrical stimulus was done by Zimmermann. The electrical stimulus, that is the electroporation method. So, when the protons are subjected to electricity, under the electricity, what will happen? The small pores will be formed, and through this pores, the DNA can enter into the cells and then transform it. So, that is called as the electroporation. In 1983, intergeneric cytoplasmic hybridization in radish and ref was done by Peltier. 1984, development of genetic fingerprinting technique for identifying the individuals by analyzing the polymorphism of DNA at the DNA sequence level was done by Alec Jeffries. In 1985, infection and transformation of leaf disc with the Agrobacterium tumefaciens, and then regeneration of the transformed plants was done by Horsch et al. And in 1986, tobacco mosaic virus resistant tobacco and tomato transgenic plants were developed using the cDNA of the coat protein gene of tobacco mosaic virus by Powell, Abel et al. In 1986, development of bioelastic gene transfer method or the gene gun method for plant transformation was done by Sanford and Klein. And in 1987, isolation of a BT gene from Bacillus thuringiensis was done by Barton et al. And in 1990, transgenic BT cotton was developed by Monsanto, and in 2000, it was approved by the Government of India for commercial production. And in 1993, Krantz and Loris produced the fertile maize plants through in vitro fertilization. So, in vitro fertilization means in the synthetic medium, the fusion of the female as well as the male gametes. So, that is the ovary as well as the fertilization of the ovary using the pollen. That is called as the in vitro fertilization, that was done in the maize plants. In 1994, plant tissue culture free plant transformation method, that is without plant tissue culture, they used the Arabidopsis floral dip method. So, Arabidopsis floral dip method means the flowers were dipped in the DNA solution, and repeated culturing of these flowers, it helped in the transformation of the flower explants, and that was again subjected to regeneration to produce the transgenic plants. That was done by Clough and Bent. In 2000, transgenic rice engineered for production of provitamin A, that is beta carotene, vitamin A, in rice endosperm was developed, and it's called as the golden rice. So that was done by Ingo Porteous and Peter Bayer. In 2016, development of somatic embryos for genetic transformation in Curcuma longa and Curcuma manga for curcuminoid biosynthesis. This was done by Piccolo-Thong et al. And in 2016. So these are the important discoveries in the case of plant tissue culture. And now we'll see about the scope of plant tissue culture. So it has got many applications. Plant tissue culture has got many applications, and first of all, it is used to identify the totipotency, nutrition, then metabolism, division of the cells, how the cells divide, how they differentiate. All these things can be studied, and also you can go for the preservation of the plant cells. And secondly, it is the morphogenesis and plant regeneration from the individual cells or tissues through the process of organogenesis and somatic embryogenesis. Organogenesis means development of shoot as well as the root directly from either directly or indirectly, and somatic embryogenesis means development of somatic embryos from the explants either directly or indirectly through callus. Then variations are generated to in vitro culture. How the variations develop? So by repeated subculturing, so you can develop the variations. So these variants are called as the somaclonal variants, and these variants may have some very important characters, and you can select the somaclonal variants for abiotic as well as for tolerance to abiotic as well as the biotic stresses. Then evolution of haploids through anther and pollen culture, including the ovule culture, haploid plants can be obtained. Then wide hybridization programs through ovule, ovary and embryo cultures to overcome both the pre-zygotic and post-zygotic sterility mechanisms. In hybridization programs, the sterility is the major problem, and this can be overcome by culturing the embryos. Then micropropagation of plant materials. So that is, multiplying the plant materials in enormous amounts, huge numbers can be obtained by plant tissue culture. Then in vitro selection of mutants tolerant to biotic as well as the abiotic stresses. Then plant genetic engineering through in vitro culture methods and DNA transfer technique. So you can produce the callus from any explants, and this callus can be used for transformation either by gene gain method or by agrobacterium tumefaciens mediated gene transfer. So these are the important applications or scope of plant tissue culture. So thus plant cell tissue culture, cells or tissues or organ, it permeates the biotechnology and cements together various aspects of science like physiology, biochemistry, genetics as well as the cell biology. So all these subjects are integrated due to the plant tissue culture. Thank you.